Modern biology identifies life through molecular interactions. It operates on the premise that biological instructions are carried out via chemical diffusion, the movement of particles through fluid environments. This model faces a significant mechanical problem: system-wide synchronization. Biological coordination occurs at speeds exceeding the physical limits of slow chemical diffusion across complex tissues. To explain this speed, biophysicists propose a parallel electrodynamic architecture. This energy network governs biological organization at the speed of electromagnetic transmission. This network functions through endogenous electromagnetic fields. It is a system of internal light, generated and maintained by the organism's own biological processes. The human organism functions as a complex optical network, using these internal fields to coordinate chemical activity across the entire body. Eastern medical traditions have long described such an energetic system. They identified these routes as meridians or channels, assuming a network of flow preceding physical symptomology. These historical maps trace precise lines across the torso and limbs, connecting specific nodes located on the surface of the skin. We see a cross-cultural consistency in these models. While Chinese maps focus on peripheral meridians, Tibetan traditions emphasize central vertical channels and radiating energy centers, or chakras. The primary scientific challenge to these maps is that anatomical dissection shows no permanent physical structures, like veins or ducts, where these meridians are drawn. Modern biophysics optical waveguides. They do not require physical walls. Instead, they are formed by the nonlinear properties of biological tissues, which can trap and guide light. To validate this waveguide model, we must examine the quantum behavior of the most abundant substance in the body: water. Liquid water is often viewed as a chaotic collection of independent molecules colliding at random. The quantum electrodynamic model, or QED, offers a different view. QED is the study of how light and matter interact. This model shows that water molecules separate into two fractions: a chaotic interstitial phase and a highly structured coherent phase. This structured phase is called a coherence domain, or CD. Within this 0.1 micrometer region, approximately 6 million molecules oscillate in perfect phase, acting as a single quantum unit. Within the CD, molecules oscillate between their ground state and an excited state at 12.06 electron volts, just below the 12.60 ionization threshold. Because of this, 13% of the electrons become quasi-free, forming a collective cloud that stores and moves charge. In bulk water, these delicate domains are constantly broken apart by thermal collisions from the surrounding chaotic molecules. For these domains to become stable and biologically useful, they must be protected from this thermal destruction, a process that happens at biological interfaces. This leads to the concept of interfacial water. This is water located within a few hundred angstroms of a biological surface, such as a cell membrane or a protein. Due to the high density of cells and macromolecules in the human body, almost all biological water is interfacial. The attraction between water molecules and these biological surfaces shields the coherence domain from disruptive thermal collisions, allowing the coherent structure to remain permanent. Once stabilized, the coherence domain acts as a trap for low-grade ambient energy from the environment. This incoming energy causes the quasi-free electrons to spin into vortices. Because these vortices are coherent, they do not suffer from internal friction. These cold vortices have long lifetimes, allowing the coherence domain to stockpile energy over extended periods of time. Biological water is an active quantum battery, capable of accumulating and storing vast amounts of energy from its surroundings. Helical structures like DNA and local magnetic fields align these internal vortices. This alignment allows their individual energy values to sum together. This process combines multiple low-frequency photons into a single high-frequency photon, effectively concentrating the stored energy. The formula for this resonance is straightforward. The system takes n photons of frequency V and converts them into 1 photon of frequency NV. Biomolecules located on the surface of the coherence domain are exposed to this concentrated high-frequency light. When the frequency of this trapped light matches the specific activation energy of a biomolecule, the energy transfers through resonance. This provides the catalyst required to initiate a specific chemical reaction. The chemical energy released by that reaction then shifts the frequency of the coherence domain, which in turn activates the next specific molecule in the biological cycle. This interplay between light and chemistry organizes cellular activity. The trapped light serves as the engine that dictates the timing and sequence of biochemical events. We can scale this model from individual domains to the connective tissues of the body. Collagen fibers act as liquid crystals, materials that maintain an ordered structure while remaining fluid. This provides a scaffold for coherence domains to align. Elongated coherence domains align end-to-end along these fibers, forming one-dimensional arrays connected by Josephson-like junctions, microscopic gaps that allow for quantum tunneling. When an acupuncture point on the skin receives a stimulus, it triggers a pulse of energy that travels down these arrays. This pulse is a soliton. It's a localized wave that maintains its shape and velocity as it moves through a nonlinear medium without losing energy. Unlike linear waves, which dissipate their energy as heat over a distance, solitons preserve their information across the biological network. The meridians of Eastern medicine are biophysically identical to these soliton waveguides. They are channels of self-trapped light that coordinate the body's internal organization. Clinical evidence of this optical system appears in the colors perceived when a person's eyes are closed in a relaxed state. These colors persist long after external light is removed, serving as a manifestation of the body's internal electromagnetic fields interacting with the visual cortex. This spectrum shows the sequence of energy upconversion. In a healthy organism, the subject visualizes a progression from low-frequency red toward high-frequency blue and violet. In a diseased state, this sequence stops early at lower frequencies, like yellow. In severe pathologies, they see only black. Under this framework, disease is defined as an energy imbalance caused by a loss of quantum coherence. This leads to jammed waveguides, or the loss of energy through biophotons, ultra-weak light particles emitted when coherence breaks down. The specific color perceived by a patient provides a direct reading of their internal water's ability to maintain phase coherence and transform energy. This suggests a therapeutic path. Regularly visualizing high-frequency colors can act as an optical massage, stimulating blocked pathways and restoring the flow of solitons. It may seem unlikely that the weak electromagnetic fields produced within the body could exert a physical influence on dense biological tissue. This is explained by the Weber-Fechner law. This law states that the biological response to a stimulus is proportional to the logarithm of that stimulus's intensity. A very small stimulus, like internal biophotons, produces a large inbound effect. This results in the reorganization of the body's internal structures, rather than energy dissipating outward. Visible light wavelengths act as precise instruments, exciting specific Josephson junctions to restore coherence without the heat damage associated with external high-intensity therapies. Internal light serves as both the catalyst for chemical activity and a diagnostic indicator of an organism's internal health and organization.